Thank you all for coming. I'm really excited to share with you what I've been working on during my PhD over the past 5.5 years. My dissertation is about developing unsupervised machine learning algorithms for a bunch of different kinds of single cell analysis. And I wanted to just first acknowledge there's a really diverse audience here with a bunch of different kinds of backgrounds. I'm really excited to have a bunch of folks from my lab and my department in the life sciences. I think there's some folks here also who come from a more mathematical, quantitative background. I'm also really happy to be able to share my work with my family, who I think does not have a lot of either of those two disciplines. And so I'm going to start off just at like a very high level to provide some context before getting into more of the details of what I've been doing. So hopefully it's not surprising for all of you to hear that our bodies are composed of many different kinds of cells. There's different cells in your brain and your muscles and your immune system and your fat that all serve very different jobs that are very specialized and important for all the different functions that are required for life. But there's a fundamental question in biology of what is it that actually makes these cells different? So we generally understand that all of the cells in the body have the same genome, so the same set of instructions for producing genes and different products. But sort of the last 70, 80 years of biology has really sort of revealed the fact that it's the way that these instructions are read, it's which genes are turned on and transcribed into RNA, and then how they are translated into proteins and the modifications on those proteins that actually define or provide the differences between, say, a neuron and a muscle cell. And so biologists are really interested in trying to figure out, well, how can we measure this? How can we figure out exactly what these differences are? And a lot of the work that I've been doing was really lucky timing, because in 2015, there was a brand new technology that was introduced, which is called Droplet-based single cell RNA sequencing. And the basic idea behind this technology is that it's possible to, in a microfluidic chamber, this is a very small device, introduce individual cells to chemistry required to measure genes that are expressed in those cells in tens of thousands of very tiny droplets that can then be collected and used for experiments that allow you to amplify and then eventually measure all of the genes that are present in each of these individual cells at really high resolution. And I actually think it's really cool to see this in action. This is probably one of the coolest videos I've seen in my PhD. This is one of those chambers. We're seeing here a single capture event. There's a cell coming in from the bottom. This is using a drop-seq chamber. I said microfluidics earlier, just to give you a sense of how small this is. Each of these droplets is about 125 microns in diameter. And for reference, a single hair from your head is about 75 microns. So we're not talking about things that are that big. It really is a feat of engineering to be able to capture tens of thousands of these droplets reliably in an experiment in a way that we can measure what is going on inside each of these cells. And the output of these experiments really produces very large data sets. So when we talk about big data in biology, especially in single cell biology, the output is these data matrices or tables. You can think about like Excel sheets, where you have each row is a different observation. So each row corresponds to a cell. And in a given experiment, you may see anywhere from 1,000 to some technologies allow for millions of cells to be observed. And then for each cell, you also have a number of features that are measured in each of them. And so for a lot of the single cell data that I'm talking about, we're looking at either the RNA expression, the amount of so each column is a gene, and we're measuring the abundance of each gene in each cell. There's also some proteomic technologies that allow you to measure protein abundance. And genes we typically think about in terms of 20,000 genes per cell. Proteins, current technologies really only allow for measurements of about 100 or so, if you're lucky, closer to 50 most often. We're not just measuring this in one sample, we're actually measuring now a few samples, sometimes actually hundreds of samples, where we want to understand what's going on. And so in preparing this talk today, I wanted to just sort of acknowledge something that I experienced at the beginning of my PhD, which is that the kinds of tools needed to analyze this data is very different from what I learned when I was studying microbiology at UMass Amherst. And I think my first experience sort of trying to understand what single cell methods how they worked is I just felt like I was being totally introduced to a very different discipline. The language was different, the notation was different, the letters were confusing. And in the interest of fairness, this is actually an excerpt from a paper that I contributed to, Fate, which I'm going to talk about a little bit later. And so it's really essential actually to use this notation to be clear about what it is that we're doing. But I know how shocking it can be to just be or confusing to just sort of be push with this immediately. So one of the things.
I just found a typo in this page the other day.
Oh
2
no!
years after it's been published.
Yeah, 2 years or something like that. I'll have to go back and take another screenshot after we finish. So one of the things that I'm really been happy to be able to work on, really proud to be able to work on, that Smitha mentioned, was this machine learning for single cell analysis workshop. This is something that I started actually with Scott Gigante here, and Smitha and Matt helped a lot with the neural network section. There's a whole bunch of TAs, a lot of people to thank here. But what we did was start to actually think about how we can teach some of the foundations of what we're working on to the biologists that actually end up using a lot of the methods that we use. And I'm really actually excited to say that as of today, you can go to this link and see all of the lectures from our most recent workshop available for free on YouTube, along with slides and coding notebooks if you want to learn more. But I'm going to start off just by giving, for everyone here who's maybe not from the quantitative side, just a very short introduction to the first part of my title, which is, unsupervised machine learning. What do we mean by unsupervised machine learning? So to start with that, I want to describe just what do we mean by machine learning in general. This is a buzzword that I think a lot of us have heard from all different kinds of sources. And there's actually a bunch of different ways that people define it. And the one that I'm going to use today is relatively simple. I think of machine learning as the process of identifying patterns in data. It's a very general thing to try to do. Patterns can be all sorts of different kinds of things. I'm going to give you a couple of quick examples right here by breaking down this field of machine learning into, I think, probably two of the biggest subfields, which are supervised and unsupervised. Probably the easiest place to start is with supervised learning. It's probably what people are the most familiar with when they think of machine learning. And the general problem is that you have a bunch of labeled data, and you have new data that you'd like to apply those same set of labels to. So I'm saying labeled data, for example, I could be talking about a bunch of images, say of fruit, where I've had an expert sit down and look at them and say, these images are of strawberries and these images are of bananas. And then the machine learning model is exposed to each of these examples in the training data and then trained to produce the known output. So you go through all of your training data, you make sure that your model can apply the right label to them. And then when you have new data come in that maybe you haven't seen before, say you've got a website and someone's uploaded a new image and no one has gone through and said, this is an image of a strawberry, it is the model now can apply this label that you've seen before to that image. However, this is not as useful right now for single cell biology because we don't have labeled samples. I showed you that image of the cell flowing through this microfluidic chip. We don't even have that video for most of the cells that we sequence. We just know that we measured, some subset of genes being on or off in that cell at certain levels. And so what we work on mostly in the Krishnasamy lab are unsupervised learning models. And the idea behind unsupervised learning is that instead of having a set of labeled training data, we have a set of data that we'd like to apply some organization to. And we're going to take some assumptions about how that data might be structured, apply it to the data, and see if we can identify patterns from it that are interesting. And so going back to this example of images of fruits, we might give the model one of our examples, and the model is going to use its inherent assumptions to produce an output that is, generally speaking, lower dimensional. So I can tell you this image is about 100 pixels by 100 pixels. And here I'm showing you just 2 coordinates at the end of the day. So we're going from 10,000 dimensions to 2. And this is just a simple XY plot that you have seen in almost any times that you're looking at sort of your X position here and your Y position here. You go through all of your data. And the idea is that the model is going to produce for every single point one representation of that data, where then you can look at it and try to find structure. So here I would say structure or patterns. I noticed that these images are sort of grouped together and these are grouped together. You might even describe them as this is one cluster of data and this is another cluster of data. And you can go in and explore it and say, well, what makes these two things different? And you might say, well, this has leaves and multiple fruits per image. This is only one. For cells, you might say, well, what genes are different between these two different groups that we're seeing are separated by our model? So this is a very sort of general way to say it, but to sort of introduce you a little bit to some notation that we're going to use later in the seminar, the way I could say this idea of labeling new data as having some function on my input data X to Y, where all values of Y are known for some samples of X. So F is just some function that I give, say, images, and it gives me labels. And I know all of the labels for some samples of X. Unsupervised learning, on the other hand, where I'm trying to organize data, I want to learn an embedding. I want that embedding to be lower dimensional. It doesn't necessarily have to be real numbers like I'm showing here. It could be clusters, but the basic idea is that instead I'm learning some function of my input data to Y, where all that I know is that my input data X is really high dimensional. It's from the high dimensional real space, and the embedding is from another set of numbers where the dimensionality of the input data is much greater than the output. And so it's really important to have some of these unsupervised learning methods for single cell data, because I was telling you before, this is really high dimensional. It's really difficult to figure out how do you analyze data when you have, say, 20,000 columns of an Excel sheet, you can imagine, to try to actually look through. It's very difficult. You need some way to organize the data to sift through it. And so today I'm going to talk to you about 3 algorithms that I've worked on during my PhD to help organize and analyze single cell data sets. And the first, all of these have been published, which I'm really excited about. First, going to talk about MELD, which is a way to actually compare different single cell data sets. I'm then going to introduce Vertex Frequency Clustering, which is actually sort of built alongside and some of the same framework as MELD, which is also a clustering method to actually identify cells that are affected by perturbation. And then finally, I'm going to switch gears a little bit, although I'll hopefully make a successful segue to this, and introduce a neural network that actually allows you to characterize heterogeneity within a single cell data set. And so to start off, to introduce MELD, I want to talk to you a little bit more. I said that unsupervised learning algorithms have some inherent assumptions that they use to organize the data. And in our lab, and especially during my PhD, the assumption that we take most commonly is something called the manifold assumption. And the idea behind the manifold assumption is that although our data might be very high dimensional, we have hundreds or not tens of thousands of genes that we're measuring in a given cell, we think that there's some underlying structure within the data, and that structure is locally Euclidean, continuous, and we believe is lower dimensional than the original data. And so to sort of show this idea, on the left, I have points that are uniformly sampled from 3 dimensions. You can imagine these are 3 different genes. You can see this is sort of just almost looks like random noise. And on the right, we have something which I hope it is at least apparent has some structure to it. You could, for example, naturally follow a path through this data, whereas it's not really clear how you do that on the left. And so on the right, we have a classic example from manifold learning, the Swiss roll. On the left, we just have uniform random noise. Those who come from the life sciences actually are probably a little bit more familiar with this idea of a low dimensional manifold than they might realize. If you've ever seen an example of Waddington's epigenetic landscape, which is actually an analogy that has been around since the 1940s, the idea that Waddington came up with was that epigenetics provided a way, a landscape upon which that guided cell differentiation akin to the way that a marble rolls down a hill. And this landscape actually is dictated by epigenetic markers at billions of positions along the genome. But at the end of the day, it might guide an individual precursor cell to only, say, 4 different states. We have this low dimensional sheet representing a much higher dimensional system, the entire genome. So there's this question now, how do we actually learn this structure? How do we represent this manifold? How do we identify that within a data set and apply that assumption to data? And one of the most fundamental things to the work that we do in the lab is using graphs. So I know a lot of times when I talk about graphs, people think of that sort of XY Cartesian plot I showed you earlier. That is also called graphs. We tend to call them plots to differentiate them from mathematical graphs, which are collections of nodes and edges. Sometimes nodes are called vertices. So on the right here, we have an example of a graph with 6 points, 6 edges, and these gray lines represent connections between them. And graphs are actually really flexible for representing a lot of different kinds of data. So you might imagine if you're thinking about the internet, you might use a graph to model the relationship between computers, where you have network connections connecting computers that are connected in the internet. If you're modeling traffic, you might have intersections as vertices and roads as edges. For social networks, hopefully it's relatively straightforward to think of people and friendships. And then for cells, what we're really interested in here are similarity relationships. So when I talk about graphs moving forward in single cell data, I'm talking about graphs where the nodes or vertices are cells and edges connect similar cells. So what does that actually look like, and how does that help us identify patterns in data? So you can think of a very simple example where you have a data set in just 2 dimensions here. This might look a little bit familiar previously. The steps to learning a graph is that you first calculate all pairwise distances between all of the points. It might be difficult looking at this to see what the underlying structure in the data is, but the key to the way that we use graphs is a function called a kernel function, which translates distances into affinities. And the key is that kernels are biased. So you can think of affinity or similarity as the inverse of distance. If something is very far from you, it's not as similar to you, especially if you think about genes, right? If it has very different gene expression, it's not a very similar cell. But kernels are very biased in that small distances tend to represent high levels of similarity, but there's a very rapid cutoff. And after some amount of time, distances between cells rapidly decay to essentially not matter. And what this says is that we only care about local distances. And so what that looks like is going back to all of these distances. Now these lines represent distance, these lines represent similarity. And what we see is that only the edges between very close together points are preserved in our graph. And hopefully looking at this representation, it's hopefully a lot clearer what the underlying structure in the data is, even than just up here. And this is a relatively simple example. There are some really cool things we can do once we've learned this graph. We can look at, for example, how you might simulate random walks from adjacent nodes over time, something called data diffusion, to learn these underlying structures. And I just want to emphasize right now that this is, I think, one of the most fundamental parts of the work that we do. And for those of you who maybe aren't familiar with these ideas of Euclidean distances and kernel functions, I really just want you to take away that right now that what we're doing is looking for individual cells. What are the cells that are transcriptionally the most similar? And we're just going to keep those relationships and say we don't really know anything more than that these 2 cells are similar. If they don't look very similar, we're not going to make any assumptions. But graphs allow us to sort of extrapolate from that to learn very complex relationships in the data. And so one of the examples of this is actually a dimensionality reduction method that it was one of the first projects I worked on as a PhD student. I contributed to, just to be clear, I'm not one of the major drivers of this effort. But this is a tool called FATE, which is a dimensionality reduction tool for single cell data. So this allows you to take a data set. This is about 30,000 cells. Each point is a cell collected from a 30-day time course. The color of the point represents the day from which the cell was sampled. And what's really cool is that although this may just look like a pretty plot, I can actually tell you if you look at the different kinds of cells here, what you can see is a progression. On the right here, we have embryonic stem cells from early on in the time course developing through many different kinds of cell states into some mature stem cell descendants that we see in the system that's being studied here. And so I just want to show that, but we can use these graphs to actually produce representations of the data that are really useful. I also want to give a quick shout out. If you're interested in this idea of dimensionality reduction and visualization, there's a really cool project I worked on. It actually turns out a lot of my PhD I worked on with Scott. You're going to see him come up a few times here, where we actually put together an interactive visualization that explains not just fate, but actually these 3 methods, I think we can safely say are all graph-based, and some other dimensionality reduction methods which I was really excited. We actually got an award for at a data visualization conference. This is freely available on GitHub right now. So far, what I've explained is that graphs can represent structure and data, and we're going to get to the single cell analysis that I've worked on very quickly, but there's one more fundamental concept I need to make sure people understand, which is the idea of a graph signal. So I showed you this picture of a graph earlier, but there's a lot of cases where we actually know things about the nodes in a graph. So for example, if we're looking at the internet and we have these computers, we know for each computer maybe how much data was uploaded in a month. Or if we're looking at traffic, how many cars went through that intersection last week, with the age of different people in a social network. And for cells, there's actually a lot of pieces of information we have. We know all of the genes that are expressed in them. We know all sorts of information about the sample conditions from which they were collected. And we might be interested in analyzing this information over the graph. And graph signal processing actually provides a really interesting framework for that. We might want to do all sorts of kinds of things like remove noise or recover missing values. We might want to try to characterize how this signal changes over a graph or identify regions with particular characteristics. If you're all familiar with the idea of signal processing or signals in general, one of the most fundamental properties that's required to start to do a lot of these tasks is to have a notion of something called frequency. So here I'm showing a few sine curves that are varying over time. And so here it should be pretty easy. Most people can intuitively say, well, this is a lower frequency oscillation than this is. We can see that the time scale is the same. And frequency is really important because it's the thing that differentiates, your microwave from the radio that's going to your car, from the Wi-Fi in your house. And for time varying signals, it's actually really easy to describe, well, really easy. It's possible to describe the frequency using a weights of sines and cosines, which provide a basis for decomposing signals. So there's this transform called the Fourier transform, which decomposes any signal within a fixed bound or any oscillating signal with regular period into a weighted sums of sines and cosines of increasing frequency. And so actually these signs here could be used as the basis to describe another set of signals. I'm not going to get too much into that because really the question that we're interested in is how do we define frequency for a graph? It's really easy to see, how many times do you hit a peak per unit time, difference frequency here, you can use these sines and cosines. What do we do for graphs? It turns out that there is an operator over the graph, a matrix, a representation of the graph that we can use called the graph Laplacian that's really useful for this. It's a difference operator, which means that if you evaluate a signal using the Laplacian at a given point, it tells you how similar is that signal compared to its adjacent points. So a high frequency signal would mean that you have very different values than adjacent nodes on the graph. It's calculated actually in a relatively straightforward manner from the degree and adjacency matrix. It's just degree minus adjacency is our Laplacian matrix. And what's nice about this is that when you eigendecompose the graph Laplacian, it gives you a really nice orthonormal basis that you can use to do a Fourier transform for signals on a graph. Just something that's going to mean a lot more to some of the more quantitative folks here. But hopefully what's pretty clear is that so this is a grid graph, and I'm showing each point in the graph colored by the weight of each of the eigenvectors of the graph Laplacian here. And so this is the first non-trivial eigenvector. What you can see is it uniformly increases across the graph. If we compare this to those signals I was showing you earlier, hopefully it's pretty clear that this is low frequency. We can then look at higher order eigenvector or higher eigenvalued eigenvectors of the Laplacian and see that things actually get what seems to map onto our notion of higher frequency, right? So there's sort of this like sinusoidal pattern we see if you look at any row here or across column or across the diagonal or across the columns, we can see some notions of frequency being represented where higher eigenvalued eigenvectors represent higher frequency signals. And what's really important here is that this gives us a way to do a Fourier transform on a graph. So we can take graph signals, do some Fourier transform into a graph spectral domain, and then do an inverse transform back into the graph. And this actually slide, I want to give a shout out to of David Schumann, a professor at Macalester College, who really sort of pioneered or popularized a lot of graph signal processing. And what's really important is that it's easier to do some things in the graph spectral domain than in the vertex domain. And I think a simple analogy for this, for the folks who maybe are not as quantitative, this is another sort of one of Schumann's analogies, is imagine that you had, say, a signal is like a bowl of guacamole. And let's say that what you wanted to do is take that guacamole and make it less spicy. It's really hard to just go through that bowl and pick out all of the jalapeno seeds to make the guacamole less spicy or more spicy, for example. But imagine you had sort of a magic way where you could decompose the guacamole into all of its component parts and easily separate out the onions from the tomatoes from the jalapeno in this separated space, go in, remove all the jalapeno, and then magically put it back together in a way that you can do over and over again. It would be much easier to make some changes to the underlying thing you're looking at, either a signal or this guacamole example, if you could go back and forth. It turns out you can't really do that with cooking, but actually you can do that with graph signals using this Fourier transform. And so with that, I'm going to get into exactly why this is useful and powerful for single cell analysis, talking about MELD, which is a project I worked on very closely with Jay, Alex in the lab, Scott, and then we had a really great scientific collaboration with the Herald lab with Annalisa, who generates some data, important data for this. So I introduced some of the ideas of graph signal processing. We're going to come back to them very shortly. And I wanted to make sure I explain that because you're going to see very quickly how that it can allow you to do some really important things for single cell analysis. So the problem that we have in single cell analysis, one of the big ones is that originally experiments started to just profile heterogeneity in a tissue, but more recently people have been using single cell techniques as a way to measure the effect of a perturbation. So this was a data set where T cells were grown either with or without antibody-coated beads and then sequenced using droplet-based single cell sequencing. And the output is 2 gene expression matrices, one for each condition, where for each cell, we know what genes are expressed and what the treatment condition was for that cell. So this is going to be our graph signal. I'm going to hint to you right now. But so we're interested in understanding what's different between these two samples. It turns out that's actually not an easy thing to do. You might think, well, okay, I've got 2 independent sets of samples from different distributions. Can I just do a simple t-test on the genes? And it turns out actually you can't. If you look at genes that we know play a role, so this antibody stimulation is supposed to activate a T-cell receptor. And we know if you look at genes that we know play a role in T-cell receptor activation, if you just compare the 2 samples and do differential expression or do it, simple statistical tests on the features between samples, you don't see a lot of differences. And just to orient you here, each point is a cell. The y-axis is expression for one of 6 genes that play a role in T-cell activation. And the red dots are the average for either the cells in the naive or in the stimulated condition. So not a lot of difference. What about those dimensionality reduction techniques I showed you earlier? Could we use that? It turns out that also is not that easy to do. Here, each point is a cell. The color, again, is the sample. And what we see is that there are some cells here. I know it's hard to see. I'm sorry, this is a color map from the published paper. If you look at this cell in green, we see that there's a cell from the naive condition that is very close to cells from the stimulated condition. And the converse is also true. There's some stimulated cells look very much like naive cells. And so, right, if they're close together here, it means that they're similar. I was talking about that idea of similarity earlier. And so this sort of hints at us that there's a problem. The effect size of these perturbations, the effect of adding these beads to stimulate these T cells, has a relatively small effect relative to the underlying heterogeneity in the biology and also the measurement noise and the technology that we're using. And it's not so shocking because it may be that activating T cells only turns on 10 or 100 out of 10,000 genes. It's relatively subtle to try to understand the difference here. So a few years ago, my PhD started realizing this problem and took a little bit of a step back to think, well, what are we actually trying to do here? And the basic way that we started to think about how these experiments are working is that we're looking at some underlying density function over the cell state space. So you may notice this looks a little bit similar to that Waddington landscape image I showed you earlier, just upside down. And so the idea is that we have some axes of cell state, and then some function that tells us for any cell state we might observe, what's the probability that we're going to observe a cell matching that configuration in an experiment. And so there's going to be some function for the control sample, and there's going to be, as a result of the perturbation, a change to that function, that data density, as a result of, say, turning on or turning off a gene or adding a drug or changing the media, something like this. What we're really doing is altering the function that we're drawing samples from. So really what we're trying to do here is understand the effect of an experiment as a change in these underlying probability densities. So you can see is that what we want to do is identify what are the cell states that are the most enriched or the most depleted as a result of a perturbation, and identify maybe if we just compare those cells that are the most affected to each other, we'll be able to get a better sense of that signature of the perturbation, as opposed to, say, including cells that we know are just completely unaffected by the perturbation. So those of you who are a little bit more quantitatively inclined might be thinking now, okay, mentioned data density, can we do density estimation? And actually, this is exactly what we're going to talk about. So when you're thinking about data that's distributed in the real numbers, it's common to use a kernel density estimate to estimate these density functions. And anyone who's used a KDE plot has done kernel density estimation. The way that this works, showing a histogram of some numbers distributed on the real line. So here, these are bins, and the height of each of these bins represents the number of data points that were sampled in the range of that bin. And kernel density estimation provides a way to localize a kernel function. And if you remember, actually, this is exactly the same kind of kernel function I was mentioning earlier when we talked about building graphs. You're actually going to find that this idea of using kernels and smoothing has a lot of different applications and a lot of different kinds of the analysis that we do. So you do is you localize a kernel to every position here, and then you sum them up. And when you look at this, what you see is an estimate that is smooth over the support, and that there are regions where, say, we didn't actually measure any data here, but we can still calculate the probability that we would have given this. And actually, I'm going to steal a slide. I realized I shouldn't have given you credit here, Smitha, but this is a slide from that Smitha made showing how you can do this. I showed you one dimension before. Now we're going to do it in 2 dimensions. We have some raw data. We're now localizing those kernel functions. Think of that smooth sort of quick drop off from density, from distance to similarity, centered at each point. And now imagine you just add up basically how many rings overlap at any given point in this space. And this might give you some contours that you can then use. So right where there was more data, sorry, you'll see the axes change here a little bit, where there's more data, we have sort of a higher contour, which gives us an estimate of more density. And we can do this continuously over the space that we have. And so this is all really great. And this gives us some estimate of the underlying data. Perfect, let's do it. Turns out you can't. Estimating density in high dimensions is incredibly challenging and requires, as the dimensionality gets large, an incredibly intractably large number of samples. We need hundreds of thousands, almost 1 million samples just to estimate density in 10 dimensions accurately to 3 significant digits. We're talking about tens of thousands of dimensions. We can't do that. But there's one trick that I was mentioning before, which is that we don't do a lot of our analysis in the ambient space, right? We have this underlying manifold assumption. And what we do instead of estimating density across the gene space is we actually estimate density over a graph approximating the manifold. And we do this using graph signal processing. I'm going to explain more about that because I think this is actually the coolest part of the work that we do. But the way that this happens is we start by creating a mass associated on the graph associated with each point using the sample signals, right? So this is like those points that we saw previously on that XY plot. We create some value associated with each cell from each sample. We then use a kernel function to smooth those signals over the graph in the graph frequency domain. And then we use this to get a density estimate of each sample that we can then compare. So to show you what this looks like, I'm going back to this example. I'm going to get rid of the T cells for a second and just abstract this to two samples, control and treatment. And the first step is that we learn a cell similarity graph. So remember I said before, we use a kernel function to build the graph. We're using a Gaussian kernel here. So here, each cell is a node and edges connect similar cells in this data set. And then what we do is we create a set of signals over the graph from these condition labels. So we might have just one column, say, of an Excel sheet that says this cell barcode was sampled from this condition. But what we do is actually translate that into a value of 0 and 1. So the idea is that cells from, for example, the treatment condition, this is the treatment signal here, have a value of 1 and everyone else is 0. It's the opposite for the control. I'm not going to show that for clarity, for brevity, but just know that there's 2 of these. And the goal is actually to get from this indicator signal an estimate of density over the graph. So this is not an incredibly hard concept to do theoretically, but actually there's a really cool way that we can do this that we do in MELD, which takes advantage of this graph Fourier transform that I talked about before. So if you remember, I had said that we have some signal defined in the vertex domain. Here, the signal is 1 for cells that are in the treatment condition and 0 elsewhere. And then what we want to get is some smooth signal at the end. So, what you could do is iteratively go around for each point and say, well, what's the value of your neighbors, and then average it, and then do that, in a bunch of for loops iteratively. But it turns out there's actually a much easier way to do it using this Fourier transform. And so the fundamental idea is that the Fourier transform allows you to go from the vertex domain into the spectral domain, where the signal is represented using coefficients of a set of a Fourier basis, which are here eigenvectors of the graph Laplacian. And then you can go back into the vertex domain to analyze it. And so the way that we do that in MELD is we start with the indicator signal in the vertex domain. We do a graph Fourier transform into what's called the graph spectral domain. So this is actually the signal from that T cell data set. This is the treatment signal from the T cell data set. What you can see is that each point here represents a coefficient associated with a different Fourier basis. So it's like the coefficients are, when I was talking about the guacamole example, it's like how much jalapeno is there, how much onion is there, how much tomato is there. Those are the coefficients. And then so that's the y-axis, and then the x-axis is just sorting them based off of frequencies, which I talked about earlier. And so more loadings on the left are low frequency, more loadings on the right is high frequency. And then we do what's called a low pass filter using, guess what, a kernel function to smooth the signal over the graph in the Fourier domain, in the graph spectral domain, and get out a low frequency signal. And hopefully what's clear, especially if you look at the y-axis here, I couldn't do them on the same scale because you wouldn't be able to see this at all. There's a lot more loadings on these low frequency eigenvectors. We've created a lower frequency signal, and it should be, and then we can easily go back into the analyze this, look at it in the graph vertex domain. And hopefully it should be a little bit clearer that this is a higher frequency signal than this, because I was talking about this idea of looking at adjacent nodes. Here, the average distance across the edges of this signal across the graph is relatively high, right? Because the distance along this edge is 1, the distance along this edge is 1, the distance along this edge is 1. But here, the maximum difference across any edge in the graph is much smaller. We're looking at like 0.3 or 0.5. So if you sum up all those differences, you actually see that this is more similar. There's lower frequency over the graph than here. Going back to our single cell data, so now we are looking at real data again. This is that T cell data set I'm showing you. I showed it in PCA before. I'm actually using FATE here, so we used in the paper. And so the output of MELD are these sample densities for each sample. And so here we have on the left the simulated sample, and on the right we have the unstimulated sample. And I know that with this color map, they actually look very similar. And I think that's actually really helpful to see, because in reality, they are very similar. There's not a ton of differences between these two samples. That's why it's hard to analyze. But what's great is that once we have this independently for each sample, we can compare them. And we can just look at the ratio between these two densities by normalizing them and get out a likelihood that a given cell would be observed in one condition relative to another. And so here, this likelihood is scaled between 0 and 1, such that likelihoods greater than 0.5 indicate a cell that is more likely to be seen in the stimulated condition than in the control condition. And so we look at this, there's actually one of these for each of the samples, and it gets a little bit more complicated if you have more than 2 conditions, although it definitely still works. But what's really nice is now it's really easy to see here that this is where there are more cells in the stimulated condition, and this is where there are more cells in the control condition. And this allows us to actually take now this discrete label that we had before, this experimental label, and then produce a smooth, continuous estimate of the effect of the perturbation for each cell. And we're actually going to get back to this data in a little bit, but just to sort of stress how broadly applicable this technique is, I'm going to tell you a really quick story. This is actually a really exciting collaboration led by Manik Kutru and Jesse Wang with Patrick Wong in the Akiko lab, Akiko Iwasaki, analyzing flow cytometry data from 168 patients infected with COVID-19 at Yale New Haven Health. Or maybe it was actually just across Yale New Haven hospital system. And for each cell, we have a measurement of a bunch of proteins present in that cell. And we also have a label. So I was talking before about the stimulated and control condition, but here we actually know whether or not the cell was sampled from an individual who succumbed to their illness and died, or someone who lived on. And so what we can start to do is actually now use MELD to figure out what are the cells that are enriched in individuals who suffered mortality from COVID versus those who survived, and identify what are the kinds of cells that are more likely to be seen in patients who survive or patients who die. And this, I think, was a really powerful analysis that they led, and actually identify a subset of T helper cells that are enriched in patients who experienced COVID mortality that they're going on and further investigating. I think this is really cool work to see. And so with this, I'm going to sort of summarize this first of 3 algorithms. The basic idea is that MELD provides a framework to quantify the effect of a perturbation, although, as I said before, it doesn't actually have to be a perturbation. It just can be the difference of label density. But the important thing is it's at each cell. And we're going to talk about why that's important in a little bit. But MELD uses a low-pass filter in the graph spectral domain to estimate density. I think this is one of the really cool contributions of MELD, is this way of doing density estimation in the graph frequency domain. And what's also really cool is there's a lot of different kinds of data that this can be applied to. It's broadly applicable. And so in the future, there's a few things I'd really like to see be developed with MELD. One of them is looking at how we can use, how we can actually in more detail describe differences between conditions. I show a lot in the MELD manuscript how you can compare cells that are different, but not necessarily like which samples are most similar to each other. I couldn't say take 2 of those COVID patients and say which are the COVID patients that have the most similar distributions very easily. I also, for statisticians here, didn't mention p-values at all. And I can't say how certain are we that that cell is actually enriched using replicates, but I really would like to. And then finally, we're always doing benchmarking in the lab, and I'd like to find more accurate ways to do that, which also allows me to plug another project I'm really excited to have also been working on with Scott, which is this, I mentioned this open, so you can go right now to openproblems.bio, which is a project that we've been leading, trying to actually do this kind of benchmarking. And I think one of the really exciting stories to come out of that is that after MELD was published, there are a few other labs that also published methods to do the same thing. And instead of just sort of competing in behind the scenes, we're actually collaborating on benchmarks together, which is really cool. And we're doing a jamboree this week, in a couple of weeks, sorry. Great. So I'm realizing a little bit short on time, so I'm gonna speed up a little bit through these next 2 methods. I was talking before about graph spectral representations of signals. And as I was working on developing MELD, one of the things that Jay and I realized was that analyzing the frequency content of signals over a graph is really interesting and actually can be used to identify populations of cells that are behaving in similar or different ways to a response of a perturbation. And this resulted in a project that we called Vertex Frequency Clustering that was published at the IEEE Data Science Workshop a couple of years ago. It turns out peer review is a little bit faster for conference papers and for publications. The basic idea behind Vertex Frequency Clustering is that if you have a sample label of cells from either a control or a treatment condition, we observed this in some data sets that there are some kinds of areas of the data where it seems like there's a cluster of cells that's just totally unaffected by a perturbation, right? You look around here and cells in this blob sort of at the top are equally likely to be observed in either condition. On the bottom, it looks like actually there's sort of a progression where we go from cells enriched in one condition through some intermediate state to cells that are enriched in the other condition. And the goal was to try to figure out how to separate them. And the way that we do that is using something called the Windowed Graph Fourier Transform, which allows us to not just look at the frequency content of the whole signal over the entire graph, but actually look at it locally in defined regions using something called a spectrogram. We then combine that with the output of MELD to identify these different kinds of clusters. The Windowed Graph Fourier Transform is also something that comes from the field of graph signal processing. The basic idea is that it extends the idea of convolutions to a graph using a kernel. Again, kernel is going to come up in almost every section here. We define windows over the graph. So you can imagine the x-axis here is just the nodes in the graph, in a graph, and the y-axis is some signal value. This is toy data here. And what we can do is convolve a input signal over the graph with a function that gives us some window that localizes the signal over the graph. So what I hope, if there's anything that's apparent, I hope that it's that you see here that above about 1200, all of the values in the signal have been zeroed out. And this allows us to only look at the values of the signal around a location of interest, which is right here, right around 1000. We're looking centered at this region. We just want to know what's the frequency of the signal around here. Though we can then start to analyze the frequency using the same set of tools from Graph Fourier Transform, the standard Graph Fourier Transform, just using a windowed version of the signal. And we can then similarly analyze the Fourier coefficients of that signal just in that region. And it turns out it's actually not easy to figure out, well, what size of the region should I consider if I want to identify these different populations of cells that are responding differently. And turns out our solution to that was we're not going to pick one, we're just going to pick a bunch and just sort of hope that it works well. And it turns out it actually really does. So we don't just look at one size window, we actually look at several different scales using something called a multi-resolution approach, where we basically just look at a few different levels and then just add up our results. And so basic algorithm here is that we're taking some kernel function, we're windowing the scale at multiple scales, we're then convolving windows to each vertex, taking the Graph Fourier transform. It turns out it's actually important to apply an activation function to that spectral representation. It basically says, does this eigenvector contribute, yes or no? And we don't care about to what degree. And then finally, we do some dimensionality reduction with PCA for speed and then perform k-means. Again, some of the more quantitative folks might recognize that this is these last two steps here are basically how spectral clustering works. If what you're looking at is the graph Laplacian, instead we're looking at a spectral representation of a signal using the graph Laplacian. And so actually there is some, we do think of this as sort of a variant of spectral clustering. But without getting too much into the details of why this is useful, a bunch of different kinds of graphs, I just want to go back to that T-cell example from the last section and say that when you apply vertex frequency clustering to that data set, if you remember, this is the region that was enriched in the stimulated condition. This is the region that was enriched in the control condition. We can then identify three different kinds of clusters, cells that were activated, intermediate, or naive, and then compare just the activated cells to the naive cells and get out a gene signature comparing these vertex frequency clustering that is more biologically relevant than if we were to either just compare the samples themselves or use other standard approaches for clustering and single cell analysis. So for VFC, really the takeaway I want people to understand here is that looking at the frequency characteristics of graph signals provides a really useful basis for clustering single cell data in the context of perturbations. And that if we look at the windowed graph Fourier transform, this allows us to not just look at how graph signals are represented over the entire graph, but at local regions. And there's a couple of interesting advances that we're looking to develop to make this faster and look at different kinds of data. Okay, last section of the talk. Give me a second, I'm just going to throw this a little sore. Okay, so the past two sections, I spent a lot of time talking about how graph representations of data and graph signals are, and graph signal processing is a really useful way to analyze how a single cell system is responding to a perturbation. One of the fundamental things that we did in MELD was take this discrete sample label and take it into a continuous representation that doesn't just say, was this cell observed in the treatment or the control condition, but actually continuously measures how likely we would be to do that. Excuse me. So a couple of years ago, actually, this is an idea that David Van Dyke, previous postdoc in the lab, now professor in cardiology at Yale, came up with, which was thinking, there's some previous research trying to do something called archetypal analysis, which has a similar flavor to it, where we're trying to take, instead of taking a discrete representation, we want to find continuous representations of data. And this is a project that he and I worked on in collaboration with Matthew Modio, Alexander Tong in the lab for this paper. And I'm actually now really excited that Arthi Venkat, who's a junior computational biology student in the lab, is going to be taking over and driving after I leave. And to really hammer home this idea about the differences between discrete and continuous representations, I think it should hopefully be somewhat familiar to people that a lot of medicine is moving away from just saying, is someone healthy or are they sick? Instead, there's been a trend towards describing a spectrum of diseases. And so you don't just, you think, that so people are somewhere in between this like healthiest state on one end and the sickest state on the other. And you're not just sort of in one class or the other. But in single cell analysis, people still really use discrete cluster characterizations. And if you notice in the Vertex Frequency Cluster section, even the method that we developed says you either are in the activated state or you're not. There's no sort of middle ground. You're not halfway between them. This is a representation of about 30,000 cell. No, this is way more than that. This is like 70,000 cells from the zebrafish embryo. And we can see a really complex branching structure here, but the data is really analyzed in discrete clusters. And so you might look, for example, at this group of cells that are blood cells. And you might want to think, well, are these cells more like blood cells than these cells? Are they farther along that differentiation path, or are these all exactly the same? Because just looking at the color here, just looking at the cluster says, everything that is red, and there's no difference between them. Another way to think about this is using something called archetypal analysis, which has actually been around since the 90s. And the idea of archetypal analysis is, instead of saying I'm going to characterize cells or points based off of if they're in one category or another, instead, the goal for describing the data should be to look for extreme states and then say how similar is each point to each of those extreme states. And one of the first applications that this was applied to gene expression data was actually a really cool paper looking at microarrays of a bunch of different primary cancer samples. Microarrays measure gene expression. They identified 4 different archetypes. And what you notice here is that they have this basal cell carcinoma archetype, and a lot of the cells that are from basal cell carcinoma samples are closer to that archetype than others. But it's not like that they're in one group or in another group in this representation. And so we think of this as a form of factor analysis, where we're representing each point as a convex combination of pure types. The first way that archetypal analysis was done is using something called principal convex Hall analysis. The way convex Hall analysis works is you try to fit a convex polytope, a simplex, to your data, just like we showed here. We want to find the smallest here tetrahedron that you can fit all of your data in. And what you want to do is represent each point as a mapping, or sorry, as a combination of those archetypes. And so the goal of archetypal analysis is actually to learn two matrices, W and H, where W is a matrix that goes from data to archetypes, and H maps from archetypes to the feature space, such that when you do this transformation, X prime W takes your data into archetypes, and then multiply that by H gives you your representation of them back in the feature space. We can learn some new representation that we can analyze the data in. This is a linear method, and I'm not going to get into, there's a lot of archetypal analysis methods that have been developed. I'm not going to get too much into them now, but one of the things that we realized working on this project was that the basic idea is that there's some function that we're trying to learn that goes from some latent space and then transforms that data in the latent space into the feature space, which is what we observe. So you can imagine this is a gene expression matrix here. And then what we want to do is learn some inverse of this function from the feature space back to the latent space. We want to be able to easily switch between this representation in the gene expression space and the representation in the archetypal space. Just you remember before we were talking about these graph Fourier transforms where we want to learn these invertible transformations. These are really fundamental properties for data analysis. So if your inverse function from the feature space to the latent space is find the simplex that most tightly fits the data in PCA space, you have principal convex Hall analysis. This then assumes that you're going back to find some linear combination of these features into the feature space. What if there's not a linear combination? What if there's not a linear function that goes from our latent space to our feature space? And I'll give you a very simple example of that. What if my latent space is a triangle and the data generative function is a Mercator projection of that triangle onto a sphere? Now, this is a relatively straightforward nonlinear function, but if I fit a simplex to this data that has 3 points, I might identify these tips as the extrema, but if I try to take a linear combination, I'm not going to get back onto this surface of the data. We ended up going to look at a kind of algorithm called neural networks, specifically an autoencoder. And unfortunately, I cannot really get into all the details of how neural networks work. If you look at the single cell workshop, there's actually 2 full days just devoted to neural networks. I highly recommend. But the basic idea of how an autoencoder works is it is a neural network specifically designed to learn 2 functions, one the inverse of the other, going from our feature space to some hidden latent representation and back, and to do this very accurately. What we did in AANet was take this standard design for an autoencoder and enforce some constraints on how this archetypal representation had to look. And the way that we did this was basically say, not only make sure you can reproduce your output, but also make sure that everything in this bottleneck layer is a convex combination of each of the nodes in that layer, such that we can then sample back out through the network. And when we get those linear combinations, find points that are back on the data manifold that we're looking at. And the reason that we can do this and find it on the manifold instead of off the manifold in the bottom here is because the neural networks can learn very complex nonlinear functions. Final data example, tying this back into the biology, we use archetypal analysis network in collaboration with data generated by Diane Krause's lab to look at tumor infiltrating lymphocytes. I talked before about T cells in context of COVID. Turns out they're really important for your immune system. They invade tumors and then attack tumor cells. The Krause lab generated expression profiles from 6,000 TILs, and then we could start to analyze them using the archetypal analysis network. I thought this is a really cool example. This is supposed to be a T cell actually attacking a cancer cell. When we apply archetypal analysis network to T cells, first of all, this is a fate representation, but the points here are the extreme states that the network observes. And the color of each point here is the expression of the gene. And so this is interferon gamma receptor 2, which we know is upregulated on naive T cells. And you can see that cells that are closer to this point express it more highly. And I want to emphasize as well, it's not like at some point there's a boundary where things are black and then yellow, rather there's a spectrum. And I think this is what's really cool, is that we see this across all different kinds of cell types, cell extrema, that archetypal analysis network identifies. I think this is actually a much more faithful representation of cellular diversity than clusters are, because it allows us to account for how expression continuously changes as a cell becomes more archetypal of one state or another. I don't want to get too much into this, but we can actually, I just want to say that we show in the paper that as you get closer to each of the archetypes, we find patterns of gene expression that continuously change in a way that matches our understanding of how that archetype works. So, conclusion of this section is just that neural networks can be used to describe a spectrum of biological states. Arthi is actually leading some really cool work. We're trying to come up with ways to make this neural network a little bit more robust and to work on some really exciting new data sets here. But with that, before I get into acknowledgements, I want to stop and take any questions. That's the end of I know we're a little bit over time, but that's the end of the research that I'm going to present today.